Hello, everyone, and welcome to today's webinar titled Flow Cytometry as a Microbiology Tool in the Development of Next-Generation Diagnostics. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Timothy Inglis, Dr. F Carson, and Dr. Kieran Mulroney from the University of Western Australia. Welcome. You may now begin your presentations.
Thank you for that introduction, Michelle. So I'll get straight into it and raise the issue of antimicrobial resistance, or AMR. Now, AMR is the silent pandemic. We're all used to the COVID pandemic now, but quietly in the background, a pandemic of antimicrobial resistance has been going on for years. At the moment, it is estimated that approximately there are 700,000 deaths a year, every year, from bacterial infections that are unable to be effectively treated by antibiotics. This is the human face of antimicrobial resistance. And it's getting worse. It is estimated that by 2050, there will be 250 million deaths from antimicrobial resistance, from infections that simply cannot be treated by the antibiotics that we have that currently take care of those infections. So that means that there will be one death every 3 seconds from drug-resistant infections, and an estimated $3 trillion will be lost in GDP. So how is clinical microbiology going to address this problem? You have to think of antibiotics as the fire extinguishers of human medicine and animal medicine as well. Much of modern medicine is only possible because we have antibiotics in the wings in case an infection happens after an intervention. For example, most of modern surgery relies on the fact that we can open the human body, operate on the part that we need to, close it up again, and as long as there is no bacterial infection that occurs because of that operation, everything works out fine. The patient is treated effectively, their issue is resolved, and everyone gets to go home. The problem with antimicrobial resistance is that if at any point during that surgery an infection is initiated, we're relying on antibiotics to treat the infection and to resolve it. Now, as bacteria become more resistant to antibiotics, we can no longer rely on the antibiotics that we've got to treat those infections effectively. So people may have knee replacement surgery, get a bacterial infection at the surgical site, and if it's treatable, that's fine. If it's not treatable, they could end up dying of a post-operative bacterial infection. We really, really need antibiotics to support the type of medicine that we've all want and that we've all become accustomed to. Now, up until now, broad-spectrum antibiotics or empirical antibiotics were the solution that was applied. Doctors would look at a surgical site or they would look at an infection and they would take their very best, their well-educated guess as to which antibiotic will work to treat that infection effectively. While they wait for laboratory data to come back, informing them exactly what type of bacterium is there and exactly what its antimicrobial susceptibility profile is, they take their best well-educated guess. and they apply a broad spectrum antibiotic that will inhibit the growth of and kill most microorganisms. But we can overuse those broad spectrum antibiotics, and each use helps feed the cycle of inducing resistance. So while it is wonderful to be able to use broad spectrum antibiotics, the more we use them, the greater the risk that we're going to induce antimicrobial resistance and lose those antibiotics. So against a background of increasing resistance to antibiotics, the approach of throwing the strongest antibiotics that we've got against each infection is going to start to fail more and more often. And there are precious few antibiotics in reserve. It's not like once we stop using all the best antibiotics that we've got, that there are a whole range of other antibiotics that we can just slip into that spot. And this untargeted use, even initially while you're waiting for laboratory results, this untargeted use is accelerating the pace at which antimicrobial resistance occurs. A concept that is widely discussed now is the concept of antimicrobial stewardship. So using the resources that we have, the antibiotics that currently work, using those resources in the best manner possible, both to treat patients now and to try and ensure that we preserve their effectiveness for future patients. And targeting antimicrobial therapy specifically to each patient and their infection is really important in that process. And that's where antimicrobial susceptibility tests come in. The faster the better. So to really target that therapy so that a doctor can take a well-educated guess as to what antibiotic will work, and patients will probably start on those antibiotics. But as soon as possible, it is very helpful for the clinician to have actual laboratory data confirming the identity of the organism causing the infection and confirming its antimicrobial susceptibility profile. So to actually improve at this process, we need to improve the tests that we have available to us. The top row on this figure is the existing practices that we have. So a sample will come to the laboratory and on that day, while the sample is going to the laboratory and the processing of that sample is beginning, the patient, if they have a serious infection, is likely to start antibiotics immediately. Meanwhile, back at the lab, over the course of the next 2 to 5 days, the organism will hopefully grow in the laboratory, be identified, and be tested against a range of antibiotics to identify which one will most effectively treat the infection. Now, most of the time, the patient gets better and the outcome is good. But there are frequently poor outcomes. Sometimes the empirical antibiotics that they were put on at the beginning of the process don't treat the infection that they have. And because it can be 2 to 5 days until you find out what the best antibiotic is, that makes it very difficult to make sure that the patient gets to the best antibiotic sooner. So how do we change that? How do we improve that? Obviously, part of a new paradigm for that is to have better methods that are more rapid and more accurate. We're probably not going to change in the short term that pattern where a patient comes in and empirical antibiotics are prescribed on the spot. I doubt that it's going to be a while before we get in front of that first dose. But what we can improve is the detection of organisms and confirmation that they are there, identifying what they are, and determining their antimicrobial susceptibility. Ideally, it would be preferable to get that done the same day that the patient has presented. So they may get a first covering dose of empirical antibiotics, but then as soon as possible, we could ideally get them to the optimal antibiotics for treating their infection. And sort of one of the holy grails of that area is same-day decision-making based on data. and it's our hope that this would lead to improved patient outcomes. So we really need rapid, culture-independent diagnostics so that we can provide clinicians with actionable results in much quicker timeframes.
Thanks, Christine. So flow cytometry is a powerful tool for research and can be used to start to address some of these new diagnostic challenges. But you need to start somewhere. And so realistically, the challenge for all of this begins long before you enter a lab and start thinking about using a flow cytometer. And so the first step in using flow cytometry to address some of these challenges is to identify the difficult biology that you're looking at. And that will start straight away with sample preparation. A lot of the time people will come to these problems and they think, okay, we've got a blood sample or we've got a urine sample, let's just put it through the flow cytometer. And I'm sure that any of the core facility managers that hear me say that have probably cringed quite terribly, because a lot of the time you don't want to do that. You need to think about what is exactly going on in that sample before you begin your development process. And that can come down to simple things like thinking about non-target particles within your sample. So if you're looking for bacteria, you've also got to think about all the other components that might be in there, the human cells, debris, chunks of tissue, all of those things that they're not diagnostically useful for what you're looking for, but could really damage the flow cytometer by clogs and those sorts of things, or they could mask your signal. And so one of the best places to start with that is to be thinking about particle identity. And the easiest way to do that is go and grab yourself a microscope. Look down the microscope at the sample you're wanting to work with and say, what can I see? Do I just see a nice clear field with a few bacteria and that's what I want to look for? Or is it murky? Is it horrible? Figure that out before you take any further steps. Because the other factor of all of this is that you're still talking about living organisms when you're talking about microbiology. And so there are going to be some very large challenges about the behavior of those organisms in those specimens that you need to be thinking about long before you pick up a pipette. And so the next phase would be to design around those challenges. So one of the best ways that you can start to identify that is, if you look at an Attune NXT as an instrument for your development, you have the ability to do accurate volumetric measurements without the need for additional counting beads or anything like that. That allows you to do things, for example, like spiking in known concentrations and looking at what your signal to noise ratios look like and all of those additional factors. And then that can help you guide with your reagent selection. One of the greatest challenges working in microbial flow is that we don't have all of the markers that the immunologists are used to working with. Antibodies are not as readily available, and when they are, the coverage across ranges and species is not as great as it could otherwise be. So you need to be thinking about which dyes you're going to use, how you're going to use them, and what that will really do to your experimental design. And the follow-on from that, obviously, is looking at your solutions and your buffers, because if we think back to the fact that we're working with living organisms, if what you're looking for in your diagnostic application or your preclinical development is something that is based on physiology and not just detection, well, you need to be sure that you're not perturbing the very phenomena that you're trying to see by using some of your conventional solutions and buffers. And then to follow from that even further is to think about the timeline. And then if you're working with a live organism, if you take a measurement and it's a single measurement, that can be fine. But if you're starting to look for sequential measurements or the workflow you've designed takes 6 hours, the difference between where your sample was at the end and at the start of those workflows it can be very, very large and very impactful. And so once you've made all these decisions, you've developed what you're going to do, the next step is jump in, hypothesize, test, and develop around it. So get to the lab and decide what it is you're going to do, make your first experiments, and then consider the techniques that you're going to use with it. Flow cytometry is very powerful, but you've got to think about all the ways that you're going to confirm what it is you're seeing with the flow and how it is you're going to be sure that what you're seeing is what you're actually assaying in that sample. So pre-calculation can help with a lot of that. And as I mentioned before, the idea of spiking in known quantities of organisms and using the powerful volumetrics of the instrument to be able to see exactly what you're measuring compared to what you expected to measure can tell you a lot without having to spend a lot of reagents and a lot of time in the laboratory. And then adapt to the data as you see it. There's no point continuing to throw, good reagents after bad, hoping that you're going to get what you want if the data that's coming out of your original assays is not lining up with what you expect it to. So what I'll do is I'm actually going to show you a real-world example of some technology that we've developed in our lab and some new assays that go with it, using the setting of peritoneal dialysis-related peritonitis. And most people will have given me a blank stare as to what I'm talking about, so I'm going to give a little background. And so the first thing you need to know is about end-stage kidney disease, which most people know about kidney disease. It is an ever-increasing problem as our population ages. More and more people are needing support for their kidneys. But end-stage disease is where you've reached the point that your kidney disease is so severe that you need some sort of renal replacement therapy for life. And when it comes to renal replacement therapy, transplantation is the goal for everyone, but not everyone can get organs, whether they don't match or they're ineligible for transplant. And so dialysis is one of the ways that you need to keep those patients supported, either waiting for transplant or if they're ineligible as ongoing care. And now peritoneal dialysis is a form that most people don't know about, though it is gaining popularity in some places, where instead of the traditional hemodialysis, where you take a patient, hook up their blood to a machine, clean it, and then put it back, peritoneal dialysis is actually performed with an indwelling surgical catheter in the abdomen, and then you can fill the abdominal cavity with a fluid that all of the things that your kidneys would normally filter will then drain into, and then you can remove it. And it's superior in a lot of ways. Patients are not tethered to a chair for their dialysis session. They can go about their life. They can do all of the things that they want to do. But there is a real risk associated with that. And that main complication is peritonitis, which is an infection of that abdominal cavity that largely comes about because you have this indwelling surgical catheter. And that complication is driving people away from it. So right now, the only way that we test is using culture-based techniques. We take the fluid that comes out and we say, does this grow an organism? Yes or no? And unfortunately, even with the best available practices, in about 20% of cases, that doesn't grow anything, even when there is clinical presentation and the observations made meet the guidelines for the diagnosis of peritonitis. The patient clearly has an infection, but 20% of cases, you've got nothing to work with. So that really needs us to look at what we're going to do, because right now, as Christine mentioned earlier, empirical antibiotic therapy is the way that it is treated. A patient presents, the clinician will look at it and say that is an infection, and then they will give very extreme antibiotic regimes because you can't take the risk that those patients are going to be fine until the lab tells you something maybe days later. The current best estimates from clinical data suggest that every hour that you go without effective antimicrobial therapy, 6.4% risk of death. That's unacceptable. So we need to start looking at better treatments. So any diagnostic that will enable those treatments and with targeted antimicrobial therapy must be both very, very rapid and very accurate. We need to be able to get to the really rapid turnaround, and we need to be able to have absolute confidence, because if you get it wrong and they get inappropriate therapy, there's a very good chance that patient would die. And then the reason we've turned to flow cytometry to start addressing some of these challenges in our development is that PCR and other nucleic acid amplification-based tests have completely failed to solve the issue. And there is an entire, lecture series on exactly why that is, but we'll see some of the reasons as we look at what we've done with flow cytometry. So what we did, in line with those early philosophy statements I made about how to do the design, is that we kept it very simple when we started. We had specimens provided under a biobank arrangement where we could start to do some of this testing, and we could start to look at what was going on in there. So we used very simple cleanup techniques. We centrifuge-based rather than chemical, because we really wanted to see if we could get some of those living organisms out of there. And then we selected 2 dyes to work with. So we're working with cyto9, which is a nucleic acid binding dye that will allow us to say, yes, that particle has DNA in it. And then also the live dead fixable violet viability stain, because that will allow us to get an understanding about whether a particle we're looking at, A, contains protein because of the way that the dye binds, and B, also will come back and say whether or not that it's alive or dead. And then we just allow those to go and then put the whole thing through the flow cytometer to get a view of what are we actually seeing. And when we did that, we had a populations that come with a great big smear that doesn't really look particularly informative. So we had to then build a hypothesis around what it is we thought we were seeing. So we looked at the very, very, higher forward scatter and side scatter and said, well, okay, the largest particles are probably human cells. So this cluster up the top is probably our human cells. And then we went, okay, there appears to be this second cluster that's down the bottom that we say, okay, that's probably where the bacterial cells are going to be sitting. And anyone that looks at that hypothesis says, okay, great, how are you going to tell us anything about that? And so we then took it and we ran some electron microscopy, and we said, actually, look, you can see there are human cells through all of this, and that was confirmed with some CD45 staining to show that, yes, those are white cells that are there. And then we also went through and we found that sample was full of bacteria as well. So it does give us some confidence to say, well, probably, that's likely what you're looking at. And that enabled us to say, well, let's start seeing what better we can do with this data to get a little more confidence in what we're seeing. And so we ran through a cleanup procedure through our gating to start making some choices about saying, well, what events are going to be the most important and which aren't. So this is using an aliquot that's been unstained first of all, and we have our profile by forward and side scatter on the left. And so we said, well, we only really care about those particles that contain nucleic acid, so let's throw in a cyto9 positive gate and say anything that is in this gate, that's positive. So once you apply that gate to an unstained sample, you'll find that, yes, there's a few little autofluorescent events left, and some of the human cells that have been left in after our cleanup procedure right up the top on that top of that axis you can see. But for the most part, we're not seeing anything that's going to be staining. So we can be reasonably confident that if we use a stained control, anything that we're seeing there is staining positive. And so this is, again, with the same presentation, but with stained controls. And you can see that once you apply that cyto9 positive gate, there really is, a couple of populations that jump out, and we'll start addressing what some of that might mean momentarily. But if you then backgate that across, you'll see that you actually have removed quite a considerable amount of this low forward scatter and low side scatter background events. But interestingly, there are still seeming to be multiple populations in this, and we'll address that a little later as well. So once we reach that point, just clean it up, make sure there's no aggregates left. Now, we designed our protocol so that we shouldn't see aggregates because we didn't want to make anyone mad by clogging a flow cytometer. But that being said, the Tune NXT is really robust and doesn't tend to clog at all, but why take a chance? So conventional side scatter height by side scatter area, just to say, okay, there's no doublets and clusters in all of this. And then we went and looked at the fixable amine violet stain. And so we see, on an unstained control, you get a little bit of that human cell stuff bleeding through again. But then if you look, you get this viable population, and then you'll get your double population up the top in your non-viable gate, where you'll see if there are dead cells, that's where they'll come in. So by the time we layer on all of those gates, once we've looked at our forward and side, our cyto9, our viability stain, we get to a place where we've got a decent cleaned up population that we can start to enumerate and start asking some questions about some of those the volumetric comparisons that we can make. How many events are in this versus that, and start to do some more powerful things with it. So the first thing that we needed to do before we started jumping onto human samples is we needed to test those assumptions. And so what we've done is we've run through a whole bunch of different microbial control strains where we've lined up the conventional spot-based agar plate counting method and the flow cytometry to be able to say, well, if we measure X number of cells by flow cytometry. Does the conventional method line up to it? And what you can see with all of this, and if you run the statistics that go with it, there is a little bit of a bias towards the flow cytometry overcounting, not in all cases, but as a general rule, that's what you'll see, which is okay as long as you understand that bias when you're moving forward. You know, it's entirely possible that there are a whole range of reasons we could be seeing that, but as long as we know what that bias looks like and the assay we've designed is internally self-consistent, the fact that it is close, and it is close, you know, it's within an order of magnitude, which for microbiology is pretty good, that's probably, you know, sufficient for us to continue downstream. So once we got to this point, we said, well, what we are calling a microbial cell by flow cytometry, using all of those indices that we've measured for the staining, lines up pretty well with what we see on an agar plate, which is our conventional gold standard evidence. So we can be pretty thrilled with that. So let's take it to processing some of these samples that are coming from the real world and see what that actually looks like. And so what we ended up seeing was, if you compare the three separate classifications, number one was a patient who has no clinical suspicion of infection, which we'll call uninfected. We have patients who have culture-negative peritonitis, where they meet those definitions for an infection, but nothing could be grown in the clinical laboratory. And then culture-positive, where you have an infection and they grew a causative organism in the laboratory. So for the uninfected, we had patients and the subjects under our ethics that were in the hospital for reasons completely unrelated to their dialysis. And so we took a sample there, and we were able to run that through and assay it, and we found that in those, we found roughly 10 to the 4 cells per mil, and which was quite startling to begin with, but hold on to that thought and we'll come back to it. And then in the culture negative, before we move on, in one of those, which is the little hat sitting on the top of that uninfected, we actually did take it to a clinical to our research laboratory, and we cultured a staph epidermidis out of it itself. I've left it in the data because I think it's an interesting observation, but technically, by the definitions, we shouldn't actually consider that not an infection. But then we start to ask some of these questions, and the uninfected and the culture negative really start to drive this home, which says, you know, we've got our viability stain in there. So if it says that the cells are viable, why can't we culture them then? Because we know that there are infections involved. We know that we've seen the evidence of them by electron microscopy and by the flow cytometry. So what's going on there? That's, you know, a bit of a head scratcher. And then loop it back to this uninfected. If we're saying that there's 10 to the 4 cells per mil in these patients, what is going on? You know, are sterile sites ever truly sterile? And so just by even trying to develop this diagnostic, we've come through and said, this is potentially something huge. It's something we need to investigate. We need to know more about the biology. And the power of that comes from not only really good design before you hit the laboratory, but also from using, you know, a high precision instrument with good internal reproducibility, so you can get this accurate cell counting that goes with it. So once we got all of this, and we've got 116 patients worth of data there, we asked the question, well, is good enough when it comes to a prediction about whether or not someone has an infection? And at our best, you know, across this 116 sample range, we found an 88% prediction of for sensitivity, saying, you know, of all of the actual infection cases, we can pick up 88% of them, and 94% specificity, which says if the new test says that this is potentially an infection, is that, you know, does that line up? And so in that case, you're seeing, you know, 6% of a potential false positive. So you got to take that back to the real world before you make any assumptions, because like I said before, a new test in this setting would have to be nearly perfect. An 88% sensitivity really doesn't sound like it's good enough until you think back to the real world context. And when I said before that if you've got 20% that are culture negative, so the existing diagnostics comes back and says, there's nothing here, that means that already we're 8% better at picking up an infection than that. So is it perfect? No. Is it better than what's currently used? Well, actually, yes. So from there, we're moving forward with preclinical trials for all of this. We want to really understand where that place in a potential workflow would sit and start to look at answering some of those questions. Really, is it really that good if you don't know the infection state before you are going into it? Can it do what it needs to do? And start to look at some of like the health economic questions. Ask, is it worth spending all this money on a new way of doing this to help patients? Now, we clearly believe that it will, and early modeling does suggest that that's the case. But if you want to start to translate some of your discoveries into real world practice, knowing that it's actually going to make a difference is everything. And then you need to be thinking about the validation of all of your analysis. You can't just take something that worked really good in a research setting in a laboratory and run with it to try to give results to clinicians and doctors. It's a terrible idea because of the risk involved to begin with, but also there are regulations that need to be adhered to. And if you were ever to think that you wanted to do that, there are very high bars that you have to meet for your regulatory validation to be able to get there. So it's one of those things that everyone wants to do better, but you do really need to think about, the steps that are taken when you're designing. So as a sneak peek, I'm not satisfied yet. I'm still designing for perhaps a second generation, and so many people familiar with flow will see the plot on the left here and see, t-SNE as a way to separate populations out in their relatedness to each other and their differences from other populations. And so we started taking some data and pushing it through and looking at how the algorithms determine these clusters. And anyone that looks at the overlay flow plot who's familiar with all of your human cell subset stuff is going to start to pick out a few shapes that they think they recognize. But what I found most interesting with this is we go back to this cluster 1 in green. That is what we are saying are microbial cells. And if you take the data from that and look at that, what it says, we say it's a microbial cell with our flow method, and then you take some control, E. coli and Staph aureus, and then you overlay it all. Actually, you'll find that population fits really, really nicely with the bacteria. So we're still working. The quest will never be over when it comes to developing better and with fancier tools, but this is where some of our near horizons will look like. So what I'll do is I'll change gears a little bit because we want to look even further down that potential pathway. So we can go, okay, yes, we can look at this and we can say whether or not there is an infection, but now we want to know what drug is going to work. And so our lab has developed what we call the flow cytometry-assisted antimicrobial susceptibility test, or FAST for short. And that has been designed rationally to fit into microbiology workflows that people are used to. Once you've got a specimen, you then, will make a McFarland standard, like is already done in research laboratories around the world. You then use an automated inoculator to inoculate an antimicrobial plate. And rather than, the traditional method, which would be 24 to 48 hours of incubation, depending on the organism, we're only exposing for 2 to 3 hours in this case. And then we will use that auto-inoculator again to add our stains and then put it through the auto-sampler on the Attune to give us, a full plate read in less than an hour. And then we have some software prototypes that will give us reports of all of the appropriate indices less than 2 minutes after that. So in a brief tour of how all of this works, once we've gone through the antimicrobial exposure and we've put it all through the flow cytometer, we get this data in the middle, which will give us our healthy, unexposed distribution. So we can say, this is what this bacterial isolate looks like as it was put through the flow cytometer with no drug insult. And then we can use some clustering and gating to be able to draw this purple bounded region on the right-hand panel, which we call the antimicrobial unexposed population, or AUP. And we're able to make some measurements both of the distribution, and we're showing forward by Cyto9 here. However, we do have other channels that are involved. And then we can also, using the volumetrics, we can say this is how many events per microliter we see. And so once we know what healthy, actively dividing cells look like, well, we can start to see, well, what is a drug going to do? So to demonstrate this quickly and powerfully, what we've got is Klebsiella pneumoniae, which is a pathogen of quite large international concern, and we're using the drug meropenem, which is one of the last drugs in an escalation pathway. So on the left-hand side, we've got ATCC 700603, which is a control susceptible strain, and then we've got K14, which is an extremely resistant clinical strain. And so in each of them, we've gone through the method, we've put the data through the flow cytometer, and we've bounded that region of the AUP. So this is where healthy cells will sit: 200 per microliter on the left, 326 per microliter on the right. This is with no meropenem. And then if we look at what happens when you add the smallest amount, you can immediately see the difference. The susceptible has dropped down to 12 per microliter and has already shifted most of the events that still exist outside that gate, whereas the resistant really hasn't changed much at all. And so we can keep pushing more drug concentrations. So at 1, we start to see these multiple populations emerging, almost all of them outside that healthy AUP region. And on the right-hand side, still not much. And so once we get to 4 milligrams per liter here, if you're not susceptible here, you're not clinically susceptible. You can't use meropenem, which is great. But what we wanted to see is, can we make the resistant strain do what the susceptible strain does if we throw enough drug at it? And actually, if you keep going, 16 on the right-hand side, 64, we start to see some movement. At 128, the population is almost entirely out of the gate. And then at 256, you start to see it. You see that hook shape that you see on the left is starting to evolve there. And that's actually where that isolate is susceptible. Meaningless from a clinical perspective, because if you put that much meropenem into a human, you'll also kill the human and not the bacteria. But that does allow us to see where we are. And so from there, we've actually found that you can do this for many, many, many strains and species of bacteria. So this is with Staph aureus. This is an MR assay, and you'll see the gentamicin and the vancomycin, which are 2 clinically relevant drugs. Both, they have, a susceptible phenotype by flow cytometry, and in the resistant, you can still see the persistence of that population. And this is cafoxetin, which is used to screen for an MR assay. And with the K pneumoniae, again, other drug classes will give you similar phenomena as you go through. So this is actually widely applicable across many, many, many drugs. I think at last count, we're at 13 classes of antimicrobials and many, many species of bacteria and fungi and other organisms of import. So for peritoneal dialysis samples, we actually went through and we tested 5 specimens of each of these different species: Klebsiella, E. coli, Pseudomonas aeruginosa, Staph aureus, and Staph epidermidis, which represent the most common infections in that setting. And what we found is that the agreement was actually very good. So we have essential agreement, which is agreement with the reference method within the resolution of the reference method, and then categorical agreement, which is what is shown in the heat map, which says if the existing test says it's sensitive, does the new test say it's sensitive? Yes. If it's resistant by the old test, is the new test say it's resistant? Yes. And you'll find that, yes, there are a few minor errors, which are listed in the blue, and then a few where the calls are not quite there. But these numbers are actually where a test needs to be at the most stringent levels of performance. And you'll actually find that the overall variance across all of the tested results is usually very, very good. Most of them are exactly the same. If we're saying the minimum inhibitory concentration of drug measured by one method is exactly the same as measured by the new method. And then a little bit of variance, but majority in agreement. And so again, you need to go through and you need to line up as many samples as you can to be certain that all of your validation is doing exactly what it needs to do, and that you're meeting those performance indices that could potentially be required if you were to look at moving the next step actually into the laboratory and getting regulatory validation for all of those things. And then health economics, again, is a huge part of all of this. If you want to run all of these new fancy things, you would need to be able to say, yes, this will make a real world impact. And preclinical studies without intervention other the place to go to for all of that. So to round us up in the summary, in a summary, we got to start with the cytometrist's law. Everyone knows it. You put garbage in, you're going to get garbage out. So you've really got to be certain. And the best way that you do that is doing those initial activities where you go through your rational design before you get to the lab. You validate everything that you're going to be doing, and you be certain with additional analyses like microscopy, electron microscopy, all of those other things that say what we're seeing by flow really does correlate with what we can see with other evidence. And then, to follow on from that, design with intent, know your problem really well. You can't just take a sample, throw it through a flow cytometer and expect that what you're going to get out on the other end is going to be great. You need to know what's in your sample. You need to know what you're looking for, and you need to have really thought through how to enable the assay that happens to make sure that your flow cytometry data is going to be the best it can be. And so we've proven that you can use flow cytometry to address these previously intractable challenges. It can be done, but it does require this extra level of rigor and real-world validation from those extra tools to be sure what you're looking at is really what's happening. And so we've demonstrated these innovative applications. We can detect infection really, really rapidly, and we can get these susceptibility profiles out within that 3 to 5 hour window, which means that whole workflow comes back to that same day testing paradigm that Christine was talking about earlier in our presentation. So flow cytometry is a powerful tool that could potentially address all of these challenges. And really, where it's coming back to is the phenotype, and we haven't labored the point in depth here, but that the power of the flow cytometry in all of this is that we're still measuring live organisms, and so we can be looking at all of those phenotypic expression as we're going through. We don't have to have either just nucleic acid and hope for the best, that we can tell what's being expressed and what isn't, and we don't have to have fixed cells and just hope that what we're seeing is going to be enough. We actually have, real-time profiling of the phenotype as things happen in the tube. And then translation and regulation is one of those things that, we don't talk about very much, and we probably should do a lot more. If you want to be able to take really, fun and interesting discoveries that you've got in the lab, and you want to make something out of them in the real world, you've got to be thinking about, how do you translate this? Where is the use case? And then regulatory validation. Everything we've shown you today is, research use only, but there is a pathway in jurisdictions around the world for really using some of these innovations, but you've got to be designing with intent to get there as you go, and you've got to be engaging with the experts to be able to say, we can do this, or actually, you've got to start again and you've got to rethink it. So all of these tools have promise, and I hopefully have shown you some demonstrations of just how powerful they can be, but there's still that step into real world translation that we want to be taking. And, we're encouraging other people to be thinking that way too. Cytometry is a great tool, but we can do more with it if we all work along the right lines. And so I'll round off there, but there's a huge number of people that we need to thank for all of this work, particularly the Translational Renal Research Group, Associate Professor Aaron Shakira, who runs that, and has been a great mentor to me as we've been developing these assays. And then through the FAST Lab, Associate Professor Tim Inglis, Christine, who spoke to you before, and then the rest of the team who have been toiling away at all of these innovations for many years now. And then, the University of Western Australia, PathWest, the Harry Perkins Institute, and Thermo Fisher Scientific, who have been gracious in allowing us to speak today and to communicate our findings to you. So at this point, I'll hand back over to our moderator.
Thank you, Dr. Carson and Dr. Mulroney, for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's go ahead and get started. Let's see, our first question is, Is there scope for the dyes and stains used in microbial flow cytometry to be further developed?
Yes, great question, and one that I'm a little bit rabidly passionate about myself. So I did mention briefly earlier about, how underprivileged poor microbiologists are when it comes to new dyes and new tools. With the changes in cell envelope that come with different strains of bacteria, we can't rely on antibodies as our primary method of detection. And quite often you will find that taking dyes out of the catalogue that were originally intended for eukaryotic, human cell use in particular, they will not behave the same with microorganisms. You'll find that, a single cellular living organism does a whole bunch of things differently than a cell within a tissue. And so, while there are many great tools out there, you do need to think about the way that they're going to act with the different physiology and the different structures that go with it. And so there's both a lot of potential in taking existing dyes off the shelf and repurposing them if you understand the microbiology and understand that the activity that you see is likely to be very different than you would expect from some of the eukaryotic stuff. But there's also probably a lot of room for development, and there's been a lot of innovation, particularly over the last 5 to 10 years, where there's a lot of new chemistry and a lot of new ways to look at these things. So I'm really excited about where the future for that may go, and I know a lot of other people are as well.
Thank you. Next question is, are there methods in environmental microbiology that could be replaced by flow cytometric methods?
So I'll take that question. I think that there are a number of methods in environmental microbiology that could be replaced with flow cytometric methods. And again, it comes down to that question of understanding the biology of the organisms that you're trying to detect, and in many cases in environmental microbiology, the organisms that you're trying to count. So by thinking very clearly about the intent of the existing tests and the biology of the organisms involved, I think it is possible to design alternative tests that use flow cytometric methods to detect and enumerate those organisms. Now, of course, you would have to navigate the regulatory sphere, which is not trivial. There are often local, municipal, state, national, and international guidelines that must be met around these questions. So it's definitely possible to do some of these things by flow cytometry. It's whether people have the enthusiasm to get them across the line at a regulatory level.
Great, thank you. Now, do you think flow cytometry will gradually infiltrate the microbiology laboratory?
I think that flow cytometry has already begun to infiltrate the microbiology laboratory, so particularly in research. In diagnostic settings, we're starting to see flow cytometry used for the processing of urine specimens. So in research, it's much easier to incorporate flow cytometry into the methods that you want to use to study a particular problem, because there isn't the regulation and the validation requirements around using the instruments and the techniques. And that sort of frees people up to explore, I guess, the applications that they may be able to use flow cytometry for. I think eventually flow cytometry will be used in a greater role in clinical microbiology laboratories, but there's a lot of work between where we are now and getting to that stage. Meanwhile, it's very easy to apply flow cytometry in research laboratories.
Thank you, Dr. Carson. Okay, we have time for one more question. Why isn't flow cytometry used more widely in diagnostic microbiology?
Well, as Kieran has articulated very clearly, the biology of the organisms involved in the kinds of tests that we need is quite complex. in a clinical microbiology laboratory, the specimens that come in, the sample matrix, they vary considerably depending on what specimen you're dealing with. Urine is very different to a peritoneal dialysis fluid, which is very different from a blood sample. And the complexity of those sample matrices, you need to consider all of that when you're designing your tests. And unless you are designing from the ground up with the intent to use it in a diagnostic application, it's very easy to take wrong turns along the way. So the potential for flow cytometry to be used more in diagnosis certainly exists. That now needs to be translated into, accurate, sensitive, specific diagnostic tests that can meet all the validation requirements. And that's not a trivial task. It's achievable, but it would require a lot of very focused, dedicated work to achieve that outcome.
Well, thank you again, Dr. Christine Carson and Dr. Kieran Mulroney, for your time today. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time.